So today we are going to discuss about a medical condition that is known as Erythroblastosis Foetalis. Now, to understand this condition, we have to understand what happens in this condition. So there are usually some cases about some people who have problem in their second pregnancy. In the first pregnancy, there is no problem and the child is delivered successfully. But during the second pregnancy, there is some issue because of which the fetus dies because of some reason. So there are many reasons to this, but one of the main important and one of the possible reason is called as Erythroblastosis Foetalis. Now, to understand this, we have to understand the concept about RH grouping. In the previous video, we have learned about RH groupings and we have identified that there are two types of RH. One is RH positive and one is RH negative. What is RH? RH is nothing but an antigen on any person or any cell. Antigen is a substance that can give the identity to the cell that yes, this cell is RH cell. If a cell, that is RBC in most of the cases, has the RH antigen, then the person is said to be RH positive, and if the person does not have RH antigen, it is said to be RH negative. This concept was discussed in the previous video, and today we are going to continue this process or this concept. So let us understand what happens in this case. Usually this condition arises when the male parent is RH positive and the female parent is RH negative. Now, what was RH positive and RH negative? We said that blood grouping is A positive, B positive, AB positive or O positive. Some people have A negative, B negative. So we understood in the previous video that A, B, AB and O come from ABO grouping and positive negative come from RH grouping. Now, if the male parent is RH positive and the female parent is RH negative, the first fetus will be RH positive because RH positive is found in 80% of the individual. It means RH positive is a dominant gene. It means wherever if there are positive and negative in an individual, the positive will be able to show its character much better than the negative one. That is why we say that RH positive is a dominant gene. So the fetus will have RH positive. Now, in the previous video, if we have also learned about a concept that a person who is having RH negative antigen, that is there is no RH antigen on them, so naturally they will not have any RH antibodies. So this female parent does not have anti-RH antibodies. Okay. Now this fetus is RH positive, this mother is RH negative, and this negative does not have any anti-RH antibodies. If the blood of the fetus mixes with the blood of the parent, then the parent can make, or then the mother can make the anti-RH antibodies. But when the female parent is pregnant, then what happens is the fetus and the mother's body is separated by placenta. It is separated by placenta. Placenta does not allow the intermixing of the blood between the fetus and the female parent. So when the blood are not mixed, it means the RBCs is also not fused. RBCs are also not met each other. So the female parent does not know that the fetus is RH positive. Everything goes on perfectly, and during the delivery of the first fetus, the umbilical cord is cut. So the doctor will cut the umbilical cord and the doctor will pull out the umbilical cord, and this umbilical cord is attached to the placenta. This umbilical cord contains the blood of the fetus that is already RH positive. So when the doctor pulls out the umbilical cord, the placenta also tears with it. So there is a blood loss from the mother's body. But since the placenta is torn, there is a good chance of the mixing of the blood of the fetus and the mother. So what happens is mixing of blood occurs. The mixing of the blood occurs. It means that the child is RH positive and the mother is RH negative. When the umbilical cord is cut, this RH positive and RH negative blood are mixed with each other. If the mother already had anti-RH antibodies, then the blood would have clotted. But since this is the first pregnancy, then what happens is this female parent does not have any antibody. So when this comes in contact with the blood of the fetus, then the mother's body starts producing, anti-RH antibodies. Now the mother's blood will produce anti-RH antibodies, and in the ABO blood grouping concept, or in the blood grouping concept, or even usual normal concept, we know that any antibody, if it is produced, it remains in the memory cell. That is how vaccines also work. That if you inject a dead or a weakened particle, the bodies will produce the antibodies against that particle's antigen, and then it will kill the antigen of the pathogen. Then later on in the future, this antibodies get stored, so that if any pathogen actually attacks the human individual, then the antibodies which are already stored inside the human, that can kill the pathogen. So that is the same concept which is used over here. It produces the RH antibodies. Now this everything which has happened, this was about the first pregnancy. So in first pregnancy there is no complication, because the fetus blood and the mother's blood was not fused during the first pregnancy. Now the complications start appearing from the second pregnancy onwards. So let us see what happens during the second pregnancy. So now the second pregnancy, the complications start to appear. Now the condition is same, that the male parent is again RH positive and the female parent is RH negative. But here the difference is that anti-RH antibodies are present. Now this time the mother's blood already has anti-RH antibodies. The fetus is RH positive, and since I've said that RH positive is a dominant gene, so fetus will always have RH positive. If any one of the parent is positive, then the fetus will definitely be RH positive. So fetus is RH positive. Now this fetus has RH antigen. So we have learned about the concept of placenta, that this is the fetus body and this is the mother blood. This is separated by placenta. Placenta does not allow the intermixing of blood, but placenta does allow the exchange of pore particles, gaseous and minute particles. Now this anti-RH antibodies, these antibodies are very small, but the anti-B or anti-A, those are large size antibodies, and these are small size antibodies. These small size antibodies can transfer through the placenta and reach the fetus. Now this fetus is having RH positive. That is, if you check the RBC of fetus, this will have RH antigen, and this antibody is anti-RH antibodies. So these antibodies start attacking this. This anti-RH antibody of the mother will start attacking the RH positive blood group of the fetus. This will result in RBC blast. This can result in anemia. This can result in severe jaundice in the fetus, and eventually this will die. So this is the actual process which happens. Now if you come to the name, this is Erythroblastosis Foetalis. So erythroblast is the RBC and cis or lysis of any substance. The lysis term means breakdown. So erythroblast is nothing but RBC and lysis is breakdown. So the RBC of the fetus, or foetalis means of the fetus. So the fetus RBC is broken down by the anti-RH antibodies of the female's body or the mother's body. So this results in the death of the fetus. So any more pregnancies in the future, like third pregnancy or fourth pregnancy, all the fetuses will die because the body of the mother has anti-RH antibodies. This is also called HNDLB. Now, this HNDLB, this stands for Haemolytic Disease of the newborn. Now, hemolytic, again the same meaning. Hemo, it means the blood cell or the cells or the hemoglobins. Lytic means lysis, so breakdown of the cell containing hemoglobin. Hemo is coming from the term hemoglobin that is present in the RBC. So hemolytic disease means the breakdown of the cells containing the hemoglobin of the newborn. This is called as Erythroblastosis Foetalis or Haemolytic Disease of newborn. Now, there is a question that is there any way to prevent this or not? So yes, there is one method of prevention. The method of prevention is that after the first pregnancy, after the delivery of the first baby, there is a mixture of the Rh positive blood of the fetus and Rh negative blood of the mother. So before the mother produces the anti-Rh antibodies, we have to inject the anti-Rh antibodies. So before the mother can produce these antibodies, the doctor has to immediately inject the anti-Rh antibodies. Now, what is the difference? In both ways, the anti-Rh antibodies are present, right? No. The thing is, if there is an antibody produced by the individual, then that antibody will be remain as memory cell, as in future also. But if you are injecting any antibody, then that is temporary. That antibody will get denatured after 120 days or 150 days or after some time. But if you inject the anti-Rh antibodies, this also do the same function, that they also clot the remaining blood of the positive fetus. So what happens is, the mother's body does not produce antibody. The mother's body will not produce the antibody. Then in the subsequent pregnancy, that is the next pregnancy, again the same cycle will repeat. So this was the condition that is called as Erythroblastosis Foetalis. Let us revise this what this is. Erythroblastosis Foetalis, that is also called as Haemolytic Disease of the newborn. This is a condition in which if the male parent is Rh positive and female parent is Rh negative, then the fetus is Rh positive because Rh positive is a dominant gene. In the first pregnancy, everything is good, nothing happens, there is no complication because the blood of the fetus and the blood of the mother is separated by the placenta. So there is no mixing of the bloods. Now, after the delivery of the first child, when the first child is delivered, the umbilical cord is cut and pulled out. When it is pulled out, the umbilical cord is joined to the placenta also tears, and there is a mixing of the Rh positive blood of the fetus and Rh negative blood of the mother. So at that time, what happens is the body of the mother starts producing anti-Rh antibodies. So this anti-Rh antibodies will be kept as memory cell for the future problems or future pregnancies. So during the second pregnancy, again the same thing happens and the fetus is Rh positive. Now this time the mother's blood already has an antibody to destroy this Rh positive antigen present in the fetus. So the placenta and the anti-Rh antibodies are very small in size. That is why they are able to cross the placental barrier between the mother and the fetus. And this anti-Rh antibodies destroy the fetus's Rh antigen, yes, that is present on the RBC. After destruction, what they do is it blasts the RBC, because of which the hemoglobin count decreases, which causes anemia, and it also causes severe jaundice in the fetus, and which ultimately results for the death of the fetus. So this is the reason why this is called as Erythroblastosis Foetalis, that is breakdown of the erythrocytes, or Haemolytic Disease of newborn, that is breakdown of the cells of the fetus which contain the hemoglobin. And this can be prevented. That how can it be prevented? It can be prevented that after the first delivery of the child, the doctors will inject the anti-Rh antibodies to the mother's blood. Now what does it do? It prevents the mother from creating the anti-Rh antibodies which might be stored in the future. So if you inject the anti-Rh antibodies, then they will destroy the cells of the fetus and they will itself get denatured after some times. Then for the subsequent pregnancies, there will be no complication and this cycle has to be repeated every time. So I hope you have enjoyed today's concept. So this was a video about Erythroblastosis Foetalis. See you next time in the next one. Thank you and goodbye.